The idea is that because of the corona situation, you were not able to do the practical part of the course yourself. And therefore my offer is or was that I will pipette the experiments and make videos for you. And this I would like to present to you now. Today I'm ready with the experiment number 2. So number 1 was the PCR genotyping of Tis7 and SKNC15 wild type or knockout material from the mice. One part of the experiment did not work, so I sent you, all of you, the repetition of this PCR reaction where was obviously something not properly pipetted by you, because using same reagents the reaction worked well. In any case, it is useful for you, I hope, because you will see how such a gel should be labeled, what is important to see, and what I expect, as I told you, that everybody will prepare a protocol where you will explain mainly the background, so the cloning strategy and the idea of this PCR genotyping, what is it good for, which reactions we use, and so on, and prepare the final table where you will list the numbers of mice which were tested and the result of their genotypes. So that was experiment number 1. Experiment number 2 is signaling, in this case Wnt signaling analysis, using material which was generated from these genetically modified mice, meaning what we are interested in is the regulation of Wnt signaling in the presence or absence of Tis7. The material which is used in this experiment are so-called mice embryonic fibroblasts. In our particular experiment today, we used only knockout MEFs, that means Tis7 negative MEFs. And what we wanted to show is that these cells have increased and top flash activity. Top flash is the reporter for Wnt signaling. And in order to confirm the effect of Tis7 on this Wnt signaling, we have transfected Tis7 DNA using plasmid vector called pcDNA 3.1, which is also tagged. And this should decrease the intensity of the signaling of the top flash reporter. Since these cells are knockout cells and they were generated from the Tis7 knockout mouse, you know already that this Tis7 knockout is actually a knock-in containing also a LAG-Z neocassette. And as a part of this LAG-Z neocassette, you have beta-galactosidase. So if you want to measure the beta-galactosidase activity, you will always find fairly strong signal. For that reason, I decided in this experiment not to use for normalization this step, not to measure the beta-galactosidase, but instead of that, to measure the protein concentration of lysates, which were used for the top flash, meaning for the firefly luciferase activity measurement. And that is the second part of the readout, which is necessary for the proper normalization of the results. So at the beginning, I would like to stress that you have already received the protocols. So these protocols are in, just a second, I will just show it to you because they complement the video, and if you have it printed out, it will be a little bit easier for you to follow my presentation. So we have a calcium phosphate precipitation mediated transfection, where is the full protocol of this experiment as if you would do it yourself. So there is all necessary information about the material which was used and about the procedure, which I will show you how it is done. So that was one part of the package which you have received. And the next, I will start with the video itself. So I will stop this. and I will open the next one, which is a movie showing the first part of the experiment. And that is shown here. So one more thing which I would like to stop is the video, because you don't need to have it. So if you remember correctly, please, if you have any questions, interrupt me, let me know that you are around and you don't understand something, then I will try to explain it right away. So the first step was seeding of cells. What you have done yourself, you know that we have these 7 knockout MEFs, and that we have seeded 150,000 cells per milliliter of medium in each well of a 24-well dish. The idea is that on the day of transfection, it is optimal to have 60 to 80% confluency of cells, because then the efficiency of transfection is highest. That means that for each cell type, cell line, one has to test the number of cells in the pilot experiments. For these MEFs, we have tested it before, and this was this number 150,000. Then cells were seeded in the full medium, which contains both FCS and also penstrep, which is the penicillin streptomycin antibiotics. And they were incubated overnight at 37 degrees. The next day, the first step which you have to do for this transfection is that you remove cells from the incubator and you check whether everything is all right with them. That means if they are not contaminated, and mainly what is the cell density. So the first thing is the microscopy. You have to have a look on several wells of your dish in order to be sure that the wells are even, that not depending on your treatment, everything will be the same. So what you see here is that, for instance, this particular well is quite confluent, but it is not completely dense. It's not 100%, and because there are still some free spaces, and that it is not contaminated, because if it would be contaminated, then you see very fastly moving small dots, which are the bacteria, for instance. So this is one of your real plates, one of the plates which was prepared by the students, and you see that the quality was good. So when you increase the magnification, if you go closer and you focus, you see nicely that there are cells which are fairly dense, but they are not 100%. So this, I would say, is around 70%. Then you see some rounded up cells. So this is always possible that you have also some dead cells around, but the number of these rounded up cells is not so high. As I said, there is no contamination visible. Important is also to focus on different parts of the well, so on the center and also to the edge of the well, because what sometimes happens is that they are not evenly distributed. So you see here empty spaces and round cells, not what we want really to see, not the nicely adhered flat cells. So this is the concrete plate, and what you see is the proper labeling. So this is important to check if you have many different plates in the incubator. These are our knockout cells, knockout 47, SKNC15++. So that means wild type SKNC15. I'm repeating it because you will need this information for your protocol. Important is also to notice that they were plated on 16s, because you will see that the transfection was done on 17s and the lysis and measurement on 19s. Next thing which you see in the very first moment is the color of the media. And now we are coming to the transfection. So what do you need for the transfection? This is the easiest technique, which is the cheapest technique. is called calcium phosphate transfection. The disadvantage of this technique is very low efficiency. This depends on the cell type naturally. So with some cell types, it's very good and the efficiency is sufficient. With these MEFs, with these particular MEFs, it's not so good. Notice this information because this is necessary for the explanation of our results. What you see is one of the reagents which are necessary for the calcium phosphate transfection, and this is this HEPES buffered saline solution. So the protocol for this solution is easy to find, and it's necessary just to have the optimal pH. This is very important when you do it yourself. It has to be tested because this is affecting very strongly the efficiency. This solution is aliquoted at -20 degrees, and each aliquot is used only once. Second important part of the system is calcium chloride, and calcium chloride is also prepared by the experimentator, aliquoted at -20, frozen, used only once. Now, the first step of the protocol, which is in your hands, which I have provided you with before, is that you have to prepare one tube with the HEPES buffered saline with 520 microliters. Why this volume? Because our experiment contains 4 different samples, and these 4 different samples are always done in triplicates. This technique, if everything works well, is very sensitive, and there are quite large differences between individual samples. For that reason, we use always triplicates, and then for the final evaluation of the results, we make the mean and standard deviation. When we have 12 samples, because it's 4 different samples times 3 is 12, we have to prepare solutions for 13. Why 13? Because these pipettes are not very accurate, and therefore it's always useful to have one as a backup, one sample. So we are calculating everything for 13 samples. In case of the HEPES buffered saline, it's 13 times 40 microliters, it's 520 microliters. And this solution we call solution A. It is also important to notice because in one step we will mix together DNA with calcium chloride together with this HEPES buffered saline, and there it's necessary to know that we have labeled this HEPES buffered saline as tube A. So 520 microliters. Then one technical detail: always when you use for work with tissue culture these pipettes, you have to clean them with 70% ethanol, this lower part of the pipettes, because although you are using autoclaved tips, it can happen that you touch something with the pipette itself, and that leads to contamination of the sample. So don't forget to do it always. Always clean the pipette with ethanol. Then we take this sterile tip and the HBS, the correct volume, which is this 520 microliters, and pipette that carefully. That means that it has to be this 520 into the tube number A. Then the second part of the protocol is the solution B. And the solution B contains basically your DNA samples mixed with calcium chloride in distilled water. So this is double distilled water, which is autoclaved, sterile for the work with tissue cultures. Here we need 556 microliters because it will be at the end A and B 1:1, but what we need is water plus calcium chloride and DNA, so that gives us this 520 altogether. Autoclaved water is also important to handle, as every solution in the tissue culture in a sterile way to clean the bottle with ethanol. And then it's important to notice that in the protocol, it's always a master mix, which we split into the following samples. So that means everything what is common for all samples comes to the master mix. You have to calculate it. And then you take out from this master mix parts which you enrich with the individual DNAs for each sample. DNAs are always kept at -20 degrees for long-term storage, but when you are doing the experiment itself, you have to thaw them and then put them on ice. Important is necessary to put the correct concentration of each sample so that you know the real concentration in your stock and you can calculate how much you have to pipette. So what you see here is the top flash. This is the reporter for Wnt signaling containing 4 TCF4 binding sites and the luciferase gene. Then we have one DNA which is called pGEMT. This is an empty plasmid DNA vector, and we use this for all control samples where we don't do overexpression of our particular DNA. And in this case, we complement the amount of DNA, so the micrograms of DNA, which we use for the transfection with T7. So this is construct with T7, and we want to simulate the situation in wild type cells by overexpression of T7, so to do this so-called rescue experiment. And we know how much we want to use of T7. And for the other sample, which is our control, we will add instead of T7 pGEMT, which is the empty plasmid vector, to have the same ratio between the DNA and the transfection reagent. The last sample which you see here is plasmid vector for expression of beta-catenin. And as you already know from my talk, beta-catenin is that molecule which is inducing Wnt signaling. So beta-catenin interacts with TCF4 and stimulates Wnt signaling. So in order to prove that our system works, that the readout of Wnt signaling pathway using top flash assay worked well, we overexpress in one sample beta-catenin. That means that we will have a sample where is no DNA T7 and no beta-catenin. That means that we have to complement the amount of these two DNAs with this empty vector DNA. This is just for explanation of these samples which you see here. So 4.5 microliters is therefore not 4.2 because this pipette you cannot adjust on 4.2, so slightly more DNA top flash. Top flash is the reporter. So always clean the pipette, as I told you, because this is the next pipette for small volumes. Then you have to take the tip, and what we add next is this reporter. We add it to the solution B, mix it properly. This is another step which is necessary to notice, so it has to be properly mixed. And then from this one, we will make 3 different samples, which is the B1, B2, and B3. B1 is that one where will be only the empty vector DNA. B2 is that one where we overexpress this 7. In this case, it's a MIC-His construct, which gives us the opportunity to detect the overexpression in Western blot using anti-MIC or anti-His tag antibodies. So if you want to prove it. So then you take a part from the sample which you have prepared and labeled B, and you put it into this B1. B1 is doubled amount because This will be used also for one sample, which will be stimulated using lithium chloride. Lithium chloride is an activator of wind signaling, so it's also our positive control to prove that the reporter system works as it should. The next sample contains this. So this B1 contains the empty vector PGMT. This is this 1.2 microliters of PGMT. PGMT is actually for bacterial expression of DNA in bacteria, not in mammalian cells, but chemically seen, it's a DNA. The reason why we use it for the control transfections is that we learned that certain type of reporters, in particular this top flash, are somehow regulated by other plasmids containing CMV promoter. So it's not a good idea to use empty plasmid vector with CMV promoter. And we tried it and empirically learned that the transfection efficiency has the same values, but the activity of the reporter is not affected when we use this PGMT for bacterial expression of proteins. So that was the sample. B1, and then you add calcium chloride to this mixture of water and DNA. Why it is necessary to add calcium chloride is to change the charge of the DNA to allow formation of the complex. So calcium chloride is positively charged, DNA negatively charged, and if you mix it, then in combination with this Hepes buffered cell line, it produces a complex which is then accepted by the cells and will get into the cells. What is necessary to notice here is that when you combine calcium chloride with DNA, you have to mix it, have to do it dropwise, because that gives the best opportunity for interaction between calcium chloride and DNA. Then you have to mix it 5 times up and down. This is written in the protocol. It's also important step because then you know that the technique is reproducible. Next part is the 140 microliters from this B, which we take out and use for the sample B2 and the second one for B3. So they contain already the reporter top flash. And in one case, we add this 7 DNA, and in the next one, we add beta-catenin. So this is from the master mix with our reporter, DNA mixed with the reporter top flash. And the next one is the last sample, which will be once again the reporter, and we will co-transfect beta-catenin. So this is the plasmid vector containing T7. You have to add 1 microliter. Mix it first. Important is that all these stock solutions are properly labeled. So you see there the concentration of the stock solution, which is 1 microgram per microliter. You add it to the mixture where you have already your reporter. And then you have to add the calcium chloride. Be careful, the volume of the calcium chloride depends on the amount of DNA. So in this case, we have lower amount of DNA That's the reason why we are using less calcium chloride, not as in the first sample. Once again, it has to be added dropwise, mixed after each step, and at the end, pipetted up and down 5 times. Last sample contains then beta-catenin. In this case, it's also approximately 1 microgram per microliter. So we add 1 microliter of this beta-catenin, clockwise, pipette, mix up and down. And then next step, calcium chloride. Once again, dropwise, and mix it. So this way we have prepared all 3 B samples which contain water, DNA, and calcium chloride. And then the second part is the solution A. So we have to wait 5 minutes, and in these 5 minutes, we prepare solution A1, which has the same volume as B1, A2 same as the B2, and A3 same volume as B3. What is in A is the hippies, which you have seen at the beginning. So for B, we need 260 microliters. For the B1, for B2 and B3 is 114 microliters. So actually the first step to put this 520 is not necessary. You can do it right away. The idea is only not to forget that what we have to do in the next following step is to add our DNA calcium chloride mixture to hippies buffered cell line and not the other way around. So therefore I have prepared 3 times A and 3 times B, and then we will do the next step, which is adding this DNA mixture to HBS. And the next part is to pipette it really always 15 times. The point is the best formation of the complex and the reproducibility. So you have to collect all your mixture of DNA calcium chloride and then add it to HBS, once again dropwise. What you can sometimes notice is that the solution will change to turbid. It will be slightly milky, and this is something positive which may or should happen. That's the formation of the complex and can be sometimes visible. So I'm showing it in details because you have to be really careful to collect all the solution which you have prepared. It has to be in 1-to-1 ratio. And here you have a small advantage to the normal course because you can see everything in detail when there are many participants in the course and only one is pipetting, the rest has to watch it and it cannot be so nicely visible as in the zooms of the video. Okay, so this is this 15 times. Mixing of the sample. This has to be done with all samples naturally. Important is that you follow the protocol. So it may be a little bit annoying that you see this step several times, but I have already cut the video so to make it a little bit faster than the reality. But you see the most important steps several times, so that gives you the feeling how it would be if you do it yourself. Next time, if you do it in reality, you will be better prepared. So here we have these mixtures. And the next step is to remove medium from our plate. So you remember that you have prepared cells always in 1 milliliter, and for the transfection itself should be 500 microliters of medium. That means that with a sterile tip, you remove from each well 500 microliters, which leaves you with 500 microliters of the full medium in each well. You don't need to change the tip because you have same type of cells in all wells. As long as you work in a clean way, you don't contaminate anything, it's okay. Then it's important to label it properly. So this is very important step. Don't forget to do it when you do the experiment yourself. As you see on the top, we have B1, 2 times 3 samples, because one will be then treated with lithium chloride with the inducer of Wnt signaling. And then we have samples B2 and B3. So now we add the mixture, once again dropwise, and important is to know how much. So it's 80 microliters of the transfection mix per well. And don't rush in this step, do it always dropwise and try to distribute the sample all over the well so that it gets the best chance to interact with all cells in your well. So step by step, this was added to all samples, B1, B2, B3. Always mixed in between. And then the plate has to be labeled when the transfection was done. So the time is important because afterwards it will be incubated, and in one part of the samples there will be replaced the medium with fresh medium where we add lithium chloride. So okay. The question is if everything was clear, if you understood what is done in this part of the experiment. Are you still around?
Yes, for me it was clear.
Yes.
Okay.
Clear.
Any questions? If there are no questions, I hope that you have received the protocol, that you have seen what has to be done. So the next step which I did was that I have added the lithium chloride. The final concentration was 20 millimolar in 3 wells. And that was our positive control for the Wnt signaling system for the readout. Next part of the experiment is 2 days later. So the cells are incubated for 2 days in an incubator 37 degrees and so on. And afterwards, the samples are lysed. And what I did was measuring of luciferase activity. And that is the next video. So I will start now the next video. So can you see now the next video?
Yes.
Okay, fine. So this is the beginning of the next procedure, which is done 2 days later. Here I put next to each other 2 different plates. So on the left side, you see not transfected cells. On the right side are transfected cells. So what is the difference between these 2 plates? What you obviously see?
The color of the medium.
Color of the medium, correct. Yellow color means that cells are growing fast. They are certainly overgrown. This pinkish red color is that it's alkalic, meaning that they are not so fastly growing. So there is an effect of the transfection, something happened with the cells. This is the plate which was transfected. Next thing is that here was added 1 day later than the lithium chloride. And on the bottom are samples which were transfected with 2 different. So this is on 19th. You see, once again, the first step is to check cells using microscopy. What you see here, very, very dense cells, which gives you a hint what you have to think about. Is it good? Is it bad? On one hand, it is good that cells are not dying, that you have no contamination, and that you have a lot of material to test. But the problem is that also most probably the efficiency of transfection would be not very high, because if you overexpress some foreign proteins in large amounts, then usually cells don't do very well and they are not growing so fast. So what you see, I have written here also on the bottom of the video in the undertitles that there are no empty spaces and that the plate is very dense up to the rim of, so you don't see any difference between the center of the well and the edge of the well. Next is necessary for repetition, reproducibility of the experiments, that you know the timing. It has to be always the same time number of hours which was used for treatments. In this case, you see sample where was no treatment, no overexpression, very dense cells. However, there are few rounded up cells as well. Always check cells at different regions, up and down. So if it's more than 1 layer, here lithium chloride treated cells, you see more of these rounded up cells, so they are not so healthy. That's the difference between these 2 samples. So there is a monolayer, and on the top are also growing these rounded up cells. Monolayer can recognize easily. These 7 transfected cells, once again, you see very few of these rounded up cells. The whole culture is confluent. There are everywhere cells, and you see these cell-cell contacts. There are no dead cells. Beta-catenin transfected cells, usually there is proliferation and increased, so there must be more samples, the culture should be more dense. Then for the luciferase assay, you need to prepare solutions. Now, for that purpose, I have sent you also a protocol. I will open it. This is this one. So here you see two buffers. One buffer is so-called assay buffer, and the other one is injection buffer. Assay buffer is prepared with your samples, and injection buffer you add to each measurement one by one, because this contains the substrate luciferin. So what we are measuring is the activity of the enzyme luciferase using substrate luciferin, and then emitted light will be measured. Because this reaction is temperature and time dependent, it's necessary to do all measurements under identical conditions. And for that reason, we don't add the substrate at the very beginning to all samples at once, because that would give a big difference between the first and last sample. We do also these buffers at room temperature. The reason for that is that until the last sample will be measured in the instrument, it takes sometimes longer time. And then the first sample would be measured at +4 degrees, but the last one at room temperature. That's the reason why we don't do this at room temperature, at 4 degrees, not on ice, but at room temperature. What else is written here? So you have to remove the medium, wash cells with PBS. You will see that lyse cells with a triton containing buffer. This is fairly high concentration of triton. And then put these samples into a 96 well special plate where they will be measured. So you take from 100 microliters of lysate 40 microliters. Why this volume? Because if you mix it with 150, this gives you 190, and then is added in the instrument 100 microliters of the injection buffer, altogether 290, and the volume, maximum volume for a 96 well dish is 300 microliters per well. Then what you see here is the composition of these stock solutions. In case you want to do it in the future, this was done like that, like it's written here. So this protocol you have received before. And now we go back to the video. So luciferase assay needs ATP and luciferin. These are prepared as aliquots and stored at -20 degrees. Always use a fresh aliquot because the activity depends on the these two reagents. And if you thaw it several times, it will lose the activity. ATP would be degraded in any case. So these are the aliquots, and then two buffers have to be prepared for this reaction. This is the assay and injection buffer using water, glycyl glycine, that you have everything in the protocol. ATP and luciferin have to be thawed very quickly, so that's the reason why I put it here in water bath at room temperature. Afterwards, you put them on ice to avoid degradation. Then ATP is added to the assay buffer. Magnesium sulfate is also to the assay buffer, and to the injection buffer, luciferin. And you can notice that it changes color to slightly yellowish. Then the next step, once you have prepared these buffers, is to get rid of the medium which you have in the wells of your dishes, and then wash cells with ice cold PBS. This is an important step because this medium would interfere with the reaction. So you dump the medium and add PBS into the plates. Then you aspirate. It's necessary to completely aspirate this PBS, because if you leave some PBS there, that would dilute your sample when you do the lysis. So this is similar step as you have done in the tissue culture. Try not to touch the cells, always do it at the edge of the well, because you don't want to lose your samples. Then lysis buffer is added, 100 microliters of this lysis buffer containing 2% Triton X100 detergent. This we do with this repeater pipette because it's fast and your cells will not dry out in the meantime. So everywhere is added 100 microliters of this lysis buffer. And then cells are lysed at room temperature, rotating on this shaker at least 5 minutes. It can be also longer. This depends on the type of cells. In between, you prepare 150 microliters of this assay buffer into this special 96 well dish. So it's a white wall reflective dish. The reason for that is that if the luciferase activity is fairly low, then it's an advantage, because these walls reflect the signal to the center of each well, and the probe of the instrument is measuring in the center from the top. Second thing is, if you have very, very strong signal, and if you would use a translucent plate, then the signal goes to the neighboring wells as well and would affect the measurement. Because of these painted colored walls, it is not possible. The only disadvantage is that these plates are fairly expensive, so we have to reuse them. This is the instrument. So this is the older generation which we use in our lab. We have now a much more modern, but the point is that you see that this instrument can measure 96 well dishes, and then has a setup for different types of measurements. So I'll go with you through the whole setup so that you can see how it is programmed, what is the protocol, and this is the software. It's called Microwin 2010, which actually is the new protocol now, a new software. The first step which we have to do is to prime with injection buffer. What means priming? The instrument itself has fairly long tubings, and they have to be clean, first of all. So for that, we use the protocol called washing. And the next step is priming. With the priming, you fill these tubings up to the injector with the injection buffer. And that provides you with same efficiency for all of your samples. So the first sample will be filled or will be added with the same injection buffer as the last one. If you wouldn't use this step, then you have either air or you have the washing solution in the first samples. So this is the first step which is necessary to do. Then you see here is the tubing, and this is a special filter where the solution comes in. It has to be properly inserted. I'm showing it only because of the detail that sometimes it's not properly fixed there and it sucks in air, and then the measurement doesn't work properly. So it has to be checked carefully every time. Then you have to select injector, because these machines have several injectors. In our case, it's the number 2. And we start with injecting the injection buffer, priming. In the meantime, you can check your samples which were rotating at room temperature if they are lysed. And that has to be complete lysis. How you can see that? If you keep this against a dark background, you see that there are such clumps of cells, so they peeled off the plate. You have to go and play a little bit against the light or against the dark background. And if you watch carefully, you will see that not all detached at this moment, because we have here this white circle visible in this well B3, these are cells which are still attached. Here are cells which are not detached, so there are no cells actually, just this one. But in all other samples, it's already floating in the lysis buffer. So you have to be always careful and check this situation, because if you are mainly working with a new type of cells, it can be dangerous if you are not properly lysing them. Then we have used 100 microliter for the lysis, and you remove 40 microliters of each sample and put it into the 96 well dish. So each sample is measured naturally separately. So we have triplicates, always 3 samples, should be the same treatment, should be more or less the same value. Here I have also written that it has to be properly homogenized, so you have to pipette it up and down several times uh, in order to get the same quality of the lysate. So either you decide to use only the liquid part, or you also pipette it as long as the cells are homogenized and you have always the identical type of lysate. Then don't forget that for each sample, a fresh tip has to be used in order to have really individual samples to calculate the mean value and not to contaminate with the neighboring samples your new sample which will be measured. So the next step is the measurement itself, where you see the algorithm. First is dispense. In this step, the machine is injecting injection buffer containing the substrate. Then there is a delay. So let's have a look. Injector number 2, volume is as written in the protocol 100 microliters. You can also change the speed of injection. We use the middle, and then it's important to notice that it's done by well. That means one sample will be injected and measured, and then comes the instrument to the next one. It's not done by rows or columns. And next part of the protocol is delay time. It's 3 seconds waiting time. That's important because the reaction has to be started and run always the same way. And the last step is the measurement itself, and this is 10 seconds. The number of counts which come from each sample are statistical values, so these differ from second to second. And if you use the measurement over a very short time, then you can have an inaccurate measurement. And for that reason, it's good to use longer time, and then you get more accurate measurement, because in 10 seconds you have better chance to have higher and lower counts. If you do it only 1 second, then you can have once a higher, once a lower count. So that's the decision for these 10 seconds. Then you have to select in the software which wells have to be measured. So they are highlighted here. Then the injector is selected as number 2, which we have filled with the injection buffer. And then the measurement can be started by insertion of the plate into the reader. So this is our plate. Then you have to give the name to the file. So this was our experiment, and you start the run. What you see here, that there are the zeros in selected wells, and then starts the measurement itself, and you will have to wait 13 seconds until the first value is there. You see here 356 counts is very, very low value. And this gives you the background, because here I have used samples from the not transfected sample. So this is from the plate which was not touched at all. And that is the background of the reaction. So in using injection buffer containing the substrate, you get this number of counts. if you have no enzyme luciferase around. Then next point is that these three values have to be more or less same, because this reflects the accuracy of your pipetting and also the accuracy of the reproducibility of your samples. So what we see here are 3 times not transfected cells, 3 times transfected, but only with the reporter. Then we have 3 times transfected with the same reporter, but treated with lithium chloride. Then we have the same, but transfected with T7. And then we have 3 times transfected with the reporter plus co-transfected with beta-catenin. So what is positive, we can see the difference between cells which contain the reporter and cells which were treated with lithium chloride. I told you that lithium chloride is inducing Wnt signaling, and you see that here we have around 1000 counts and here only 400. And then the next step is this 7 co-transfection, which should decrease the number of counts. Unfortunately, this experiment was not very efficient. Why not? Because you see that the values are very, very low. So if the background is around 300 and we see here only 400 where we have the reporter, the difference is too small. It should be in thousands and in hundreds of thousands and in millions, and this is not enough. On the other hand, what is positive is that the system as such works, because we see nice induction when you compare these 3 samples with these 3 using lithium chloride. And also, what is even more important from the biological point of view, if we overexpress beta-catenin, which is the main player in the Wnt signaling pathway, you see also an increase. So the top flash reporter in our hands worked. What is not working well is the transfection efficiency using this technique. To say frankly, until now, I have always used different technique, and this is called lipofection, using lipofectamine, a commercial product, instead of calcium phosphate, and with that it worked much, much better. In this case, I wanted to test whether it's possible to use this particular MEVs using calcium phosphate technique. Obviously, it was not the best idea because the efficiency is very low. The details are once again what I have explained now in my own words, is written on the film as well. So triplicates are similar, and here you have these 4 different groups. Next step is the normalization. So I told you that in this particular case, because these samples, these cells contain the beta-galactosidase activity, that would not reflect the transfection efficiency. And therefore, I decided to do a different normalization, and that is on the total protein amount per sample. And for that purpose, I have used so-called Bradford assay, where you have to mix water, 49 microliters in this case, 150 microliters of the Bradford reagent, which you can see here. So it's Comasi containing reagent, and that's a commercial product, and 1 microliter of each sample of our lysate. Why this small volume? Because the problem is that this lysate buffer containing Triton X100 in fairly high concentration, 2% of Triton X100, interferes with this reaction. So it's also not optimal normalization procedure. If for the future, I have to think about a different technique, how to measure the protein concentration to avoid the interference of the lysate buffer. But as a matter of principle, you can see that after 10 minutes of incubation, there was an obvious change in color from this brownish to blue. And then it is used for the measurement of the adsorbance using the same instrument, but in this case, the algorithm is for adsorbance, and it is at 620 nanometers. Optimally, it should be 595, but the problem is that this particular instrument has only few filters, and the closest filter to this wavelength was 620. So that's the reason why we decided to use this filter. There you don't need to add anything, you just have to select the wells which have to be measured. Just a second. Okay. Yeah, so this is the proper filter which has to be selected. 620. Energy of the light which is used for the emission is 17,000 lumen, and counting time is 1 second, so it's very fast technique. Why I'm showing this? Because you have several options how to program the machine, and this is important to use the proper energy and wavelengths. Okay, so here I have selected the samples, and here are the results, where you can see that the differences were really very minor. There were a few samples where the concentration of protein, total protein, was slightly lower, but in principle it was always the same. So we had same number of cells, and the transfection itself didn't harm them. So we had more or less same number of living cells at the moment when we prepared the lysate, and that's the reason why the normalization would not affect the results that much. But there are different options depending on what you are transfecting, how you are treating the cells, and then the changes can be much higher, bigger, and therefore it's necessary always to do the normalization, not to use only firefly luciferase values, but to normalize it either on number of cells or protein concentration, or the best way is on the transfection efficiency using an independent technique which is not regulated by the treatment, so which would give you the information about number of cells which were positively transfected. So this is all what I wanted to show you. You have seen now within 1 hour the procedure which would take you 2 days. I hope that you have understood what is done and how it is done, and you will be able to do similar experiment on your own. So what I expect from you now is that you will have a look on the raw data which I have prepared for you, and they are shown here. So can you see everything?
Yes.
Fine. So what I have prepared here are the names of groups of samples. These are not transfected cells at all. This is transfected with top flash reporter only. Then this is transfected with top flash reporter and treated with lithium chloride. So this is not treated, this is treated. Then these are transfected with the reporter plus with D7. And the last sample is transfected with reporter plus beta-catenin. And then you have triplicates. FL1 stands for firefly luciferase number 1, number 2, number 3. And protein is the protein concentration in the sample number 1, number 2, and number 3. Here are the values. So what you see here are just the adsorbance values for the protein concentration and number of light units for the firefly luciferase. So no micrograms, no microliters or anything like that. What you have to do is you always divide firefly's luciferase value with the protein concentration value, and you will get a number. You will do this for sample number 1, number 2, number 3. And then you calculate the mean of this number 1, number 2, and number 3, and standard deviation. At the end, you will prepare a graph showing the difference between these 4 samples. You don't need to use this sample because this is not interesting. That would just screw up the whole figure. What you need is to compare everything what contains the reporter and control versus lithium chloride, T7, and beta-catenin overexpression. And you will show the ratio between firefly luciferase representing top flash activity, meaning Wnt signaling, and protein concentration. So this divided by protein concentration, which will normalize the amount of sample which was used for all measurements. So the final graph should be with mean and standard deviation for all 4 samples. This is the normal way how it is presented. So all what I want from you is that you will prepare this graph and you will properly label it X and Y axis, and then you will write what we can see there. And then you can also write a short results section and mainly discussion what was the result of the experiment as I explained to you now. So what worked, what did not work, what we have seen, what we have not seen, and then your suggestion what should be done in the next experiment, next following experiment. I hope that this is clear to you. For me, it's important that you understand the system which you have used, that we go back to the mice, that you know with which genes you have worked, what had happened. That means that it's a knockout mouse for which gene, what is known from the literature about this gene and its relevance to the Wnt signaling pathway. Then about the top flash assay, what is the principle of this assay. You don't need to repeat anything about pipetting, about volumes, about amounts and so on. This is not important. You have to learn the biology mainly, and then presentation of your data. So what I want from you is a proper graph with proper labeling and then discussion to this graph. Is this clear? Did you understand me? Is there any question? Okay, so I see 1, 2, 3 persons understood, know what is going on. If you have any question, please ask me now, either through the chat or directly through your microphones. I'm here to answer your questions because you didn't have the chance to do the experiments. Okay, alles klar.
I have a question, sorry.
Okay.
So about the Excel file, so the raw data, we have to do the mean of the triplicates, the standard deviation, and then put on a graph the 4 samples to compare them, right?
Exactly, yeah. I have this graph, but I didn't send it to you because this is your task. You have to do it, you have to learn it.
But in the end, it should be one graph for the whole experiment.
Exactly.
Okay.
Yeah.
Thank you.
Then concerning the next following experiment, number 3 was you have seen me killing the animal. And the reason for that was that this was a T7 knockout mouse containing LAG-Z neocassette. And as I explained you in my talks, the LAG-Z is very good for localization of expression of the gene of interest, in our case T7. And what can be done with that is that you can identify organs, tissues, and cells where at the particular moment during development or in adult animal, this gene of interest, in our case T7, is expressed. From this material, you have seen that I've isolated different organs. These were embedded, then they were cut into sections. Sections were labeled. I am preparing another video where you will see these few steps, how it is done. And then we have already prepared microscopical images of several different organs. Each one of you will get few of these pictures. They are good, meaning the quality and the protocol experiment worked very nicely. Labeling is nice. What is not so ideal is that we don't have any counter staining. That means that the structures which you can see there are not so easy to identify, except for instance, small intestines for the jejunum, where you can see nicely the crypt and villus. And that is something what I have shown you also in my talk, so that you will be able to recognize. For everything else, if you are interested enough, you should try to find in some literature, atlas and so on, what can be the description of the image which you see there. And you have to write a protocol, a comment on these images, on the procedure, and mainly on the principle of detection. That means this cloning strategy, what is in the construct, how it is detected, what is the technique, and so on. I will provide you with these images, and also I will make the new video, finalize it, but at the moment I have no time to do it because I have to run other course. And the experiment number 4 is isolation of RNA, production of cDNA, and real-time PCR measurement of expression of a downstream target gene, which is in our case DLK1, which I have explained you in my talk, affecting adipocyte differentiation. There I have isolated RNA. Once again, I have filmed the whole procedure. Then I will do cDNA and real-time PCR, and you will get the results. But this is not finished yet, so that will take me some time. For that reason, I decided to send to all of you the material for the journal club. Journal club, as I said, will take one day, only one day. It will be 7th of December, where we will have all talks in this one day. And the idea is that each talk has to take 20 minutes, and then you should give questions, all of you, because what I would like is some interest from your side in the topic, and that you can present only if you give questions. So at the very end, you will get a note for that, it's mark for that, but that depends not only on your presentation, but also on your activity during these presentations. The topics are application of these mouse genetically modified mice in different fields as models for different diseases. So I find it quite interesting. I have tried to select relatively recent papers and also recent techniques. So what I expect from you is not to present all figures, all pictures, all data which are in each paper, but the most important, most relevant ones, to show that you understand how the mouse was generated, which technique was used, how it was prepared, and then also what is the relevance to the disease, and to show which experiment provides us with some useful data using this system, these genetically modified mice, for understanding new aspects of the disease. So this you have to do until 7th of December, and then you will present it. Who will present when? This you should decide yourself. Basically, everybody should be around. We will make 2 sessions, and as I said, 20 minutes plus discussion, so approximately half an hour for each person. It should not take us very long time, and I hope that you will be able to prepare it until then. I will now concentrate on a different course, different program. I can't finish these 2 experiments, which I promised to you now this week. But once I'm done, I will provide you with the films. I will explain it in a similar way as I did today. And then you will get also the data, and you will write the protocols. So this can take some time, and therefore I want to be fair, and I don't want to get from you the protocols within 30 days now, but only at the moment when I will present you with all data. From that moment, then 30 days should be enough. So you can prepare some parts of the protocol right away, and some you will have to wait for the new set of data. Is this clear?
Yes.
Yes.
Okay, fine.
I just have one question about the seminar. Will it be online or in
Yes.
presence?
No.
Online.
Online with the Zoom because everybody has access. So I hope that this will be fine. And the date was set already a long time ago. So keep this day free for that purpose, please.
Okay, thank you.
Okay. Did all of you get these papers? Can you open the PDF file? Can you see everything?
Yes.
Yes?
No problems.
No.
Good. So then I hope that everything is clear. Thank you for your attention. And that was it for today.
Thank
Bye-bye.
you.
Thank you.
Welcome.
Bye.
Thank you.
Bye.
Bye.